which will touch on these coloration stuff. But before that, I would like to thank the organizers, Axel and Christoph, especially for inviting me to this interesting seminar series. I hope some of you at least heard was in the audience of the previous talks, because I found them very exciting. And I also hope that you would enjoy some of my stuff. Okay, so I intended to give a vague topic with this slide. It's not. Okay. So we are a member of a laboratory called Insect Evolution and Genomics at the China Agricultural University. We have 4 researchers with different backgrounds. For example, in insects evolution, genomics, bioinformatics, microbiology, and evolution and molecular biology. And I have to say, by no means that we are experts on bee behavior or neurobiology. What I'll be presenting today are some of our recent developments in honeybee evolution and adaptation, especially focusing on Apis cerana. But some of the contents will have something to do with behavior. So if that's what you're looking for, bear with me. I promise I will present something about it. And of course, we have a fairly decent group of brilliant students. I believe we have more than 25 now, currently still in the lab. And some of them recently defended their thesis, but new students are coming in. So we more or less maintain a similar size group. And my students are working on different projects, including honeybee evolution and pollination and general biology. I don't think I have to convince this audience that honeybees are important ecological function servers. But I do like to point out that the 2 major services that they provide, namely pollination and honeybee production, are both related to the 2 major food kinds that they consume, which is pollen and honey, or nectar, if you like. And this has everything to do with honeybee evolution and adaptation. And some of these philosophy will be reflected in my talk. So among many questions, one of the main questions being asked in my lab is how have Asian honeybees became adapted to temperate climate? So why bother, right? Of all the interesting questions we could ask, it turns out the temperate actually matters in agriculture. And in fact, most of the world's food is produced in temperature zones, and which is also the regions that benefit the most from animal pollinators. Among all extant honeybee species, or the species from the genus Apis, there are only 2 species, Apis mellifera and Apis cerana, both of which belong to the sublineage cavity nesting honeybee. These are the only 2 species that manage to expand their original range to the temperate region. And I think it's not a coincidence that these 2 species are the most important pollinators to the human beings. I understand that the origin of the genus Apis remain debatable. There are a lot of theories about it, but it is relatively well perceived that the common ancestor of all the extant honeybee species came from tropic regions. For example, for Apis mellifera, there are still parallel theories about their origin, but the 2 leading theories include an African origin or a Asian tropic origin. The figure on the right was modified from a recent publication in Science Advances, which touches on the issue of the origin of the Western honeybee, Apis mellifera. Basically, they showed the evidence and conclusions that the species actually originated in Asian in the Asian area. The Asian honeybee or Apis cerana has a very different story. The species is mostly confined to the northern and southeastern and the eastern and southeastern and southern part of China, of Asia, and their evolutionary history and distribution range are mostly shaped by multiple rounds of glacial movements. We recently published a paper in 2020 using whole genome information to try to understand the evolution of the species. This evolution of the mainland lineage of the species. And the mainland lineage has a fairly large geographic distribution range, which is also the only lineage that managed to conquer the temperate region. And based on our results, we first used the genomic information to identify population structure of the lineage. Basically, what we found was the populations could be identified with the populations within China could be divided into 7 subspecies level division. With if you let me turn on the if you look at this figure, all the dots in gray colors, this belong to a large central population, and all the colorful dots belong to the different subspecies or local populations, if like. But their genetic division from the central population is large enough to be called a subspecies based on the criteria in the Western honeybee. So this is a fairly interesting distribution pattern because you don't typically see this kind of subspecies division within the species. But perhaps the most interesting part is when we look at the evolutionary history or the population dynamics of the history, and we concluded that the species has a very interesting evolutionary history where all the peripheral subspecies actually derived independently from the same ancestral central population. And to make it easier to understand, we've been calling the model centrifugal diversification model. The important take-home message here is that all these colorful peripheral subspecies or peripheral populations were the results of independent diversification events. So this is a very, very important evolutionary framework because all of our ongoing research are based on this model. So now we have this evolutionary model built, we could start approaching to some of the interesting questions about temperate adaptation of the species. Actually, I think for all animal species, there are 2 outstanding questions that the animal have to solve before they expand to a new habitat. And these are whether or not it is being well fed, or how do they keep them warm enough, right? So this is actually the two parts I'll be presenting today. So for the first part, we have to identify the exact problem for them to expand to the temperate region. what kind of food challenge that these bees are facing when they expand their range, right? Was it the diversity or kind of food that they managed to have access to? It is very unlikely so, because we all knew that the Apis species are all typical generalist pollinators, meaning that they can have access, they can consume all sorts of different flower resources, right? And this was actually confirmed by our own study. This was a study that we carried out in one of the typical Apis cerana habitats, which happened to be the, this was based in Sichuan province in the middle of China, which is also a typical natural habitat for the giant panda bears. And basically, we sequenced the honeybee bee bread and trying to understand what they have been collecting, right? So the results show that within 2 months, the colony has been collecting more than a few dozens of different flowers. And not only that, they could not only collect what we call major nectar plants, and these plants actually have very wide distribution range across China. But they also could use what we call minor nectar plants. And most of these have very scattered distribution. So these shows some very localized characters. So basically the bees are using whatever they could use. And in many cases, they managed to collect local flowers, helps them to survive in between the major flowering season of the major nectar plants. So it does seem that the honeybees were not challenged by the diversity of food. So what is it, right? What is it that has been causing problems? We actually knew the answer because we knew that the temperate honeybees will have to survive winter. And actually, honeybees are one of the very few insects that could do it that way. They do not hibernate. They do not hibernate. So they survive winter by staying alive. And how did they do that? Apparently they did that by forming a bee ball, and all the individual bees kept vibrating and generating heat, so that the temperature difference between the core of the hive and the ambient temperature could be as high as 40 to 50 degrees. And this is a very amazing adaptation. But at a cost, the whole colony will have to consume somewhere around 30 to 40 kilograms of honey for a single colony to survive winter. And of course, this data was based on Apis mellifera. We don't have this kind of data for Apis cerana, but it would be somewhat similar. So it became immediately apparent that the ability for the colony to collect sufficient nectar before winter is very important, which is a critical ability for them to survive winter, right? Now, here comes the challenging part. The temperate bees will not only have to collect sufficient nectar, they will have to do that in a very short time, because in the habitats in northern China or in the mountainous region of China, they're basically dealing with flowering season of as short as 2 to 3 months. So they have to collect enough honey in 3 months for a large colony to survive winter. The end results of this adaptation is amazing, because if you look at the production trace of all these local strains of honeybee, you immediately recognize that all these northern honeybees and mountain honeybees actually have a very strong nectar collection ability, especially when compared to those from the tropical region. How did they manage to do that, right? Of course, it has to be a very complicated mechanism, but perhaps the most parsimonious solution to this would be for the colony to simply allocate more foragers to collect nectar and less foragers to collect pollen. And in the end, the collective colony yields will have more nectar or honey, if you like, for the whole colony. And this might not be as crazy as it sounds, because we already knew that the honeybees has task divisions in foraging. It turns out that the nurse bees has shown variations in their sensitivity to sucrose. And this variation is correlated to foraging task division. So basically, for the nurse bees that has low sensitivity to sucrose, they tended to become nectar forager when they're aged, and vice versa. But for those with high sensitivity to sucrose, they tended to become pollen foragers. And we also know the foraging capacity are heritable. So we could imagine a scenario where selection would simply work on the genotypes that could produce more nectar collectors. And so the question became, so what genes are responsible for sugar sensitivity at the individual level, right? And we actually have several existing studies reporting on some of the possible candidates for responsible for sugar sensitivity. But for us, we have a very interesting model, which allows us to look at what happened in the actual evolutionary history of the Apis cerana. So basically, we have shown that the peripheral subspecies were all the results of independent diversification events. And now we can assume that the genes involved in foraging division or sucrose sensitivity are probably have been repeatedly selected, or someone would call it gene reuse. So this is actually what we saw in our real data. In the 2020 paper, we screened for all the genes that showed selection signals, and there was a small, among all the selected genes, there's a small proportion of them are being shared by at least 2 independent subspecies or peripheral populations. And among all these genes, there's one particular gene that went extreme. This particular gene are being selected in almost all peripheral subspecies. And we actually use some 3 different to screen the genes and their selection, and it was actually shown selected for this particular subspecies in 2 of the 3 methods. So this gene is immediately highlighted in our analysis. I've shown its name here, called LKR, local kidney receptor, but actually but actually when we started, we didn't really know the gene's name because it was not even properly annotated in honeybee genome. So we had to look at the sequence characters very carefully, and we had to construct a gene tree using similar sequences. And in the end, we managed to identify that the gene actually belonged to the gene family LKR. Even though this particular gene is not well studied in the honeybee, it was actually a star gene in a number of model animals, including human, mouse, Drosophila, and mosquito, and worms. And for all the functions that we know of, they are more or less related to feeding. So basically, it determines whether the animal wants to feed, or what kind of food does it like. And someone even call it the weight loss gene. Okay, so it probably will have something to do with feeding. And so to understand what exactly it does in the honeybees, we carry out some molecular work. So first we measure the expression level of the gene in different body parts and in different castes, different divisions of worker divisions. So the results show that the gene is more significantly expressed in both antennae and brain, but also it was more highly expressed in the forager bees when compared to the nurse bees. So these results basically in support of the gene function being related to feeding. And we also measured the expression level of the real foraging task divisions. So basically what we found was the expression level was significantly higher in pollen foragers when compared to a nectar forager. So this was also in what we would expect based on previous theory. And finally, we carried out a gene knockdown. So we did LKR-RNAi using a nanocarrier-based system. So basically, the gene knockdown significantly reduced the expression level of the gene in the brain, I think. And perhaps most importantly, the knocking down of the gene has apparently reduced sucrose sensitivity, which is what we would expect. So basically, with this work, we managed to show that LKR is directly relevant to sucrose sensitivity, and actually it can modulate the level of sensitivity to sucrose, which could be in turn translated into foraging task division. And the variation in this gene has presumably came from the different fathers. So we know that the honeybees has an extreme polyandry system where a virgin queen would mate with multiple drones from different colonies. So that system actually creates a possibility that there could be a high level of variations for a particular gene within the same colony. And if that were the case, that might serve as the basis for natural selection to work, right? And so basically there's a whole hypothesis built based on our findings from this work. And apparently this is a very complicated work, and we are still carrying out additional tests to figure out whether the LKR is actually responsible for task division in the foragers. And we have experiments designed to test their behavior changes, and we have experiments trying to understand their molecular mechanisms. But I have to say that for the behavior part, we have encountered some issues. Basically, the Apis cerana is more difficult to manipulate in behavioral experiments. We're still trying to understand what causes the problems, and we have only recently had some better luck in conducting the behavior tests. For the sake of time, I don't have to. I don't think I'll have the time to go into all the details. But if you're interested in learning our failure, I'll be happy to discuss this with you after my talk. All right. So So this is, I think this is a good point to stop my stop my not talk for questions. If there's any.
Does anyone has a question?
Otherwise, I would have a question.
Christoph, yeah, go.
Sure.
So when looking at individual adaptation to colder climates, one of the things that that's very interesting that you often find is that in colder climates, individuals of the same species are larger, kind of following this Bergmann's rule. And I was just wondering if you would find the same looking at colony level, that colony sizes depending on how cold it is and how far north it is, because I would assume that this affects their ability to keep warm, for example, in winter.
That is a very good question. I don't have an ideal answer to that, unfortunately, because there are very limited study on Apis cerana on that front. But I do have one support, one relevant support to this question. If you look at this bright yellow dots here, these are actually the only tropical subspecies of the mainland Apis cerana, the Hainan subspecies, and these guys were known to have very small colonies and weak ability to collect nectar, but a very strong ability to collect pollen.
Okay.
And these guys are very easy to escort and to divide their colonies. So they basically maintain a typical colony of somewhere around 2 frames if you're using that kind of nest. So this is a fairly small population size, a colony size compared to the typical temperate ones.
Okay, yeah, that sounds interesting.
Okay, there are 2 questions in the chat. Christoph, do you want to read them?
Yes, so one question or request is to talk more about gene reuse. I'm not sure if this refers to explaining what that means or if there's a specific question about that.
Okay, so by gene reuse, I mean, this is more or less a term that people use trying to make the communication easier, but it could be vague. By that, basically, what we meant was in independent evolutionary events, if a particular gene was found selected in these parallel events, then we call this gene as being reused. So I think in a layman term, we could put it this way: the honeybee tended to use the same set of genes in different locations, so that they could become adapted to various of local conditions. So basically, this is I use this as a alternative for repeated selection.
Okay. Then we have another question by Garrett Otis. And the question is, what are your thoughts about scientific names for unique bee populations defined on the basis of genetic differences, both at the species and subspecies levels?
Mm-hmm. Hi, Gar. This is an excellent question. We are actually trying to write up something which will touches on this issue. Basically, before I became involved in honeybees, I actually hated to work on subspecies or any lineages that involves human judgments. Well, that's not true because species is also involves human judgments, but you know what I mean. But for the honeybees, it actually makes the making of infra-species names actually has a meaning in applications. For example, people would be hoping to identify subspecies or even local strains. For me, I mean, it's to minimize artificial judgments. My feeling is that we need to consider the evolutionary history of the local populations, and also consider their distinctiveness in both genetics and morphology and biology. So if you're thinking if we could simply name subspecies based on genetic variation, then my answer is no. But I would say that is actually one of the most important criteria for making this call. I hope I answered your question.
We have also a question by Margarita.
Yes, can you hear me?
Yes.
Okay, so the gene that is involved in sucrose sensitivity, which drives in turn nectar holding preference versus pollen holding preference, has big implications for reproductive physiology. And I wonder if the social structure of bees, of this subspecies, which is hinged on reproductive physiology, is somehow different between different populations, whether there is a higher proportion of selfish workers, for example, that reproduce despite having a queen in certain populations. I would expect this to be the case in the pollen holding versus nectar holding populations, but I wonder if this is indeed the case.
Okay, excellent suggestions or questions. I don't have the answer, unfortunately. I would really love to have the real data for local populations, which was actually our plan after we published the paper. But then there came the COVID-19 and everything just stopped. Okay, and yeah, we would actually love to understand what it really looks like in the natural populations. I've been writing grants on that, so basically that was part of it. Well, not particularly on selfish workers, but more on the actual task division within each colony.
Have you happened to dissect workers of diverse populations and looked at their ovaries in any way?
No.
Okay, thank you.
I would suggest that we continue, and if you have questions about the first part, then we can always come back at the end.
All right. So now I'm moving to the second part of my talk. And for this part, none of this has been published.